I think it's time to talk about some very serious news. On Monday, the 26th of November, Chinese scientist He Jiankui made a public statement about the results of his team's most recent research. And let me just say that it's probably the most controversial thing I've ever personally seen in the science community. This stuff is off the charts crazy, and I can fully understand why it's so controversial, and it sets an incredible precedent that I don't really think our civilization is ready for. Do I have your morbid curiosity yet? Good. I'm going to explain what the researchers did, and then I'll explain the response from the scientific community, and then I'll give you my thoughts on the matter. Okay, here we go. He Jiankui led a research team that used the CRISPR gene editing tools to edit the genome of human embryos. They then attempted to implant the embryos and bring them to term. And just earlier this month, as a result of this project, twin girls were born. Professor Jiankui and his team have claimed that their alterations to the embryo have made the newborn girls intrinsically resistant to the human immunodeficiency virus, or HIV. The experiment involved 7 couples who were seeking fertility treatment. The men in each couple were HIV positive, and they had previously attempted to conceive while taking HIV-suppressing drugs, so as to avoid passing the disease onto their children. Professor Jiankui offered the couples a different way to have a healthy child, and from here you can see the context in which the experiment was performed. From these 7 couples, they generated embryos to study and to edit. 16 embryos were edited, and of those, 11 of them were suitable enough to be implanted into the wombs of prospective mothers. So far, there has only been 1 successful pregnancy, which has led to the birth of the twin girls, Nana and Lulu. Although, it's unlikely that the project will continue, as the university that employed Jiankui has suspended him without pay, and they've made a statement describing their response to the research as being, quote, deeply shocked. He Jiankui is also currently under investigation by medical ethics boards and all of their lawyers, who are prowling for any Chinese law or regulation that he may have violated. But despite this, Professor Jiankui has made multiple media appearances, where he tries to focus on the positive aspects of his research. In response to the huge controversy, he said that he understands the concern, but, quote, I believe families need this technology, and I'm willing to take the criticism for them, unquote. In his interviews, he describes the specific alterations that they made, saying, quote, when Lulu and Nana were just a single cell, this surgery removed a doorway through which HIV entered to infect people. No gene was changed except the one to prevent HIV infection, unquote. Because the newborn twins were apparently healthy, and because they underwent immediate genetic screening that didn't find any obvious mistakes, Professor Jiankui confirmed that this, quote, verified the gene surgery worked safely, unquote. Okay, so to be a little more specific, the lone alteration was made to a gene called CCR5. The CCR5 gene encodes a chemokine receptor that's expressed on T cells and macrophages, which work in the immune system. This receptor is normally meant for receiving signals involved in regulating inflammatory responses and chemokine responses. But unfortunately, this receptor can also bind to HIV. The virus uses this receptor as a doorway to bind to and then enter the cell, which leads to an infection. By knocking out the CCR5 gene, these T cells and macrophages don't express the chemokine receptor, and this will prevent the HIV from binding and infecting the cell. This alteration offers a strong intrinsic resistance against the human immunodeficiency virus. The newborn twin girls are HIV negative. For now, it seemed as if the experiment worked more or less flawlessly, and in terms of being a game changer, this is being compared to the 1978 birth of Louise Brown, who was the first baby to be created through in vitro fertilization. And now, we come to the response from the scientific community. In all of this uproar, I think the most prominent voice belongs to Dr. Jennifer Doudna, a professor of chemistry and molecular and cell biology at UC Berkeley, who's also a co-inventor of the CRISPR-Cas9 technology that Professor Jin Kui used in his experiment. Dr. Doudna and her closest research colleagues have some of the most well-informed and relevant opinions about this kind of stuff, so their words carry a lot of weight. Dr. Doudna said, quote, "It is imperative that the scientists responsible for this work fully explain their break from the global consensus that application of CRISPR-Cas9 for human germline editing should not proceed at the present time. It is important for the public to consider the following points. The clinical report has not been published in the peer-reviewed scientific literature. Because the data have not been peer-reviewed, the fidelity of the gene editing process cannot be evaluated. The work, as described to date, reinforces the urgent need to confine the use of gene editing in human embryos to cases where a clear unmet medical need exists, and where no other medical approach is a viable option, as recommended by the National Academy of Sciences. It is essential that this news not detract from the many important clinical efforts to use CRISPR technology to treat and cure disease in humans and in children. Public and transparent discussion of the many uses of genome editing technology must continue, as is happening over the next 3 days at the Human Genome Editing Summit in Hong Kong." Unquote. There's some critical information here that everyone needs to consider. Professor Jin Kui's work has not been published, and it has not been peer-reviewed. The in-depth mechanisms and processes of their experiment have yet to be scrutinized by outside witnesses, by independent observers. Essentially, Professor Jin Kui's claims cannot be verified at the current time. It's quite possible that future developments in this story might reveal a twist. They could have botched their procedure and not actually performed any alterations, which would be humiliating and career suicide. They could be lying, either about their process or their results, or both, which would be infuriating and career suicide. The only way Professor Jin Kui gets out of this with his reputation and his career even remotely intact is if he's being completely truthful. The experiment was done properly and it actually worked as they intended it to work, and the twin girls grow up with no health complications and they never contract HIV. The stakes are pretty high. Now, as Dr. Doudna mentioned, there's an upcoming event in Hong Kong, the second International Summit on Human Genome Editing, where Professor Jin Kui's research will be formally presented, and where Dr. Doudna herself will be moderating a panel on public engagement efforts. That'll happen at 11:30 a.m. local time in Hong Kong, so if you want to see the scientists themselves talk about all of this stuff, you can go ahead and check it out. The U.S. National Academies of Sciences, Engineering, and Medicine, which includes the Organizing Committee on Reported Human Embryo Genome Editing, released a public statement on Monday in response to the news of the birth of the twins. They cited the standards for acceptable genome editing in humans as determined by a 2018 report from the Nuffield Council on Bioethics in the United Kingdom and from the U.S. National Academy's own 2017 report. They cautioned that they cannot determine if Professor Jin Kui's clinical protocols conformed to those standards. These responses from some of the bigger players are pretty neutral, simply laying out the facts that the work has not yet been verified. But other organizations and individual researchers are giving more urgent and more caustic opinions. For example, many scientists who were involved in the summit meeting in Hong Kong have almost immediately denounced He Jin Kui's research. Jeffrey Kahn is a bioethicist at Johns Hopkins Berman Institute of Bioethics, and he's another one of the researchers going to this upcoming summit. He said, quote, "If it's true as reported, then it's an extremely premature and questionable experiment in creating genetically modified children." Unquote. Criticism has also been levied at the Chinese researchers for the possibility that the couples involved in their experiment, the people who produced the embryos, did not fully understand what they were opting into. They were told that the project was an AIDS vaccine development program, which isn't exactly a clear and unambiguous description of what they actually did. Professor Jin Kui defended the study participants, saying that while they weren't professional ethicists, they, quote, are as much authorities on what is correct and what is wrong because it's their life on the line, unquote. However, he conceded that if the experiment caused any unwanted side effects, or harm, or even death, he said, quote, I would feel the same pain as they do, and it's going to be my own responsibility, unquote. I have to butt in really quick and disagree when he says that the parents are just as much authorities on what's correct and what's wrong because it's their life on the line. It is not their life on the line. Their life is not on the line at all. We're talking about their children's life. And the children, by nature of being an embryo, cannot consent or voice their lack of consent to be a part of such an experiment. And when the experiment involves an alteration to their genome, using technology that's not exactly error-proof, it's pretty easy to see why there's such an ethical quandary here. Scientists have also criticized the researchers for choosing to do this in the first place, as opposed to using pre-existing methods of cleaning sperm of any potential HIV before fertilizing the eggs through a more traditional in vitro method. These pre-existing methods are cheaper, they're safer, and they don't carry the risk of cancers or diseases that might manifest later in life. Dr. Jennifer Doudna said, quote, if this was done to avoid HIV infection, there are alternative ways to prevent infection that are already effective. Why would you use this instead of an already established approach? Unquote. The response to He Jinkui's work from other scientists in China has been largely negative. A letter signed by 120 scientists included ominous quotes, such as, a Pandora's box has been opened. We still might have a glimmer of hope to close it before it's too late, unquote. And, quote, this is a huge blow to the global reputation and development of Chinese science, especially in the field of biomedical research. It is extremely unfair to most scholars in China who are diligent in research and innovation, unquote. The letter also explicitly said that there was, quote, a huge risk in any attempt to directly perform human embryo transformation and attempting to produce a baby, unquote. One of the Chinese scientists who signed this letter was Yang Zhengyang, a professor at Fudan University, who described the experiment as, quote, very dangerous. Dr. Eric Topol heads the Scripps Research Translational Institute in California, and he described Professor Jinkui's research as being, quote, far too premature. He said, quote, we're dealing with the operating instructions of a human being. It's a big deal, unquote. Professor Peter Broad from King's College London said, quote, it was inevitable that someone trying to reach the limelight would likely try this. However, it is often better to be safe than to be first, unquote. Shukra Mitalipov is a scientist at the Oregon Health and Science University in Portland. He was the first person to report using CRISPR to successfully edit human embryos, except he didn't implant them in a uterus. He never planned on actually bringing those edited embryos to term. And about this research in China, Dr. Mitalipov said, quote, it is premature at this stage of technology, unquote. Genetics expert Dr. Yalda Jamshidi of St. George's University of London said, quote, whether the results stand up to scrutiny or not, we need as a society to think hard and fast about when and where we are willing to take the risks that come with any new therapeutic treatment, particularly ones that could affect future generations, unquote. Remember that bit about affecting future generations, because that's important, and I'll touch on that in just a few minutes. At the Michigan Institute of Technology, Fang Zhang is a researcher who's been described as a CRISPR pioneer. He said, quote, although I appreciate the global threat posed by HIV, at this stage, the risks of editing embryos to knock out CCR5 seem to outweigh the potential benefits, unquote. He also discussed how knocking out the CCR5 gene means that the immune cells can't use this receptor anymore to engage in the normal, healthy immunological functions that it's supposed to engage in. Zhang said it will, quote, likely render a person much more susceptible for West Nile virus, unquote. Oxford ethics professor Julian Savaleska was strongly opposed to the research and said, quote, "Gene editing itself is experimental and is still associated with off-target mutations, capable of causing genetic problems early and later in life, including the development of cancer," unquote. He described the research in China as, quote, "indefensible," and, quote, "monstrous." Marcy Darnavosky is the executive director of the Center for Genetics and Society, which is a watchdog group that keeps track of these kinds of experiments. Mrs. Darnavosky said, quote, "If true, this amounts to unethical and reckless experimentation on human beings, and a grave abuse of human rights. Throwing open the door to a society of genetic haves and have-nots undermines our chances for a fair and just future," unquote. The elephant in the room during all of this controversy is, of course, as Mrs. Darnavosky pointed out, the risk of designer babies, of genetic haves and have-nots. I talked about this problem extensively in my special episode titled Evolution is Not Social Darwinism, Part 3, Gene Editing, Designer Babies, and the Future of Eugenics. I've included a link to that episode in the description section below, so if you want to check it out, you can click on it down there. But to paraphrase, designer babies will essentially take the huge problem of wealth inequality and manifest that into human biology, to the point where it will shape our civilizations and our evolution as a species. This is not a hyperbolic exaggeration. This is explicitly why this issue is so controversial. Professor Jin Kuei edited the germline, which is the copy of the DNA that'll get passed on to the next generation. Your somatic cells, like the cells in the skin on your arm or the cells in your eyeball, can be selectively edited with gene therapies with no problem. These cells are already differentiated, and they won't produce anything but more skin cells or eye cells with the genetic alteration that you put in them. But when you alter the DNA that's going to be incorporated into gametes, you're messing with the germline, with the germ cells from which all the cells that compose the rest of the organism's body are derived. So Professor Jin Kuei and his research team altered an embryo, which is a single cell that will divide over and over and over again to generate the cells that will compose the mature organism. If this embryonic genome is altered, then that alteration will be present in every cell of the mature organism's body, including the gonad tissue, which produces more gametes. These gametes, these eggs and sperm, will also carry the genetic alteration, which will then be present in any child that they create. If we're talking about selectively edited genes, we're naturally going to be talking about alterations that improve health. That's the whole point of modern medicine. That's the whole point of human gene editing. Babies that have been extensively edited will have a lot of optimally healthy genes, of optimally healthy alleles, so presumably they'll grow into healthy adults. Now, if we're talking about curing genetic diseases, and only doing that, then the ethical and practical consequences will be minimal. We'll just be increasing the proportion of people who don't suffer from genetic diseases, or people who are resistant to a particular pathogen, as is the case with Professor Jin Kuei's experiment. But there's a subtle difference between curing a genetic disease and inducing genetic resistance to an outside pathogen. Curing the genetic disease simply involves fixing the offending genes by either removing them or replacing them with healthy alleles. Deliberately inducing a genetic resistance to a pathogen is a much more active alteration. What Professor Jin Kuei did was knock out a gene to remove the infection vector of HIV, which I would argue is kind of in a gray area between silencing a gene involved in a genetic disease and outright installing a new gene to offer a novel trait. I talked a lot about the problems with outright installing new genes in that episode that I mentioned, but as for what Jin Kuei did, I don't think it comes quite to that level. I don't think knocking out a pre-existing gene runs the same ethical and practical risks as unrestricted allele shopping does. Theoretically, this alteration mechanism doesn't run the risk of creating a caste of designer elites. It's much more benign in the sense that it's just quieting a pre-existing gene. However, All of this theorizing can and will change the instant the twin girls show any kind of negative health effect that's caused by or associated with the knockout of the CCR5 gene. The problem comes from the fact that the CRISPR-Cas9 technology is kind of sloppy, and it's known to cause unintentional deletions and alterations. I think that this is a much more pressing area of concern, and you heard that many of the researchers think the technology is still too primitive and inaccurate to risk using it on human embryos. In that episode, I discussed these risks, and I explored the worst-case scenario, or the ethical nightmare of gene editing, where an embryo that's been treated with CRISPR-Cas9 suffers damage to its genome, and this produces a twisted, malformed newborn, or a newborn that suffers from some kind of immune or neurological or metabolic disorder, or a sickly child that doesn't live past puberty, or an adult who suffers from long-term ailments due to the genetic tampering. This is the kind of outcome that's totally unacceptable, and it's the primary reason why Jin Kui is receiving so much criticism for taking these risks. And then there's the whole Pandora's box thing. Now the whole world knows that you can edit embryonic genomes and create live human babies. The precedent has been set. The door is open. We very possibly might find ourselves stumbling into a bioengineering arms race, where the benefits of unrestricted genetic modification become too good to pass over. Without going into too much detail, if we do find ourselves in this bioengineering arms race, the first things that are going to appear are going to be genetically modified soldiers and genetic discrimination, like what's depicted in the movie Gattaca. The worst case scenario is that the gene modification becomes so widespread and so sophisticated that the ability or the inability to pay to modify your child ends up creating a biological caste system. The people who are literally designed to be smarter and healthier and better would naturally outcompete those who weren't altered. They would outcompete them socially, economically, and intellectually. This biological caste system would be created not through the natural randomness of independent assortment and just the luck of the draw as to what genes you get. It would be created by differences in wealth and differences in the capacity to pay to have these modifications. This wealth-based genetic difference would compound generation over generation. And at the very extreme end, there's the risk of creating a new genetic aristocracy, where kings and dynasties of the past had to claim the legitimacy to rule through a mandate of heaven or a blessing of God. Those in the modern genetic aristocracy can point to their objectively superior genes to legitimize social stratification and inequality. This is a dark, dystopian future that we should really try hard to avoid. It's not surprising that Professor Jin Kui's research sparked such criticism, when it's apparent to all that it's a big step down the road that we're pretty sure we don't want to go down. Alright, so those are my thoughts. What do you think about this? Do you agree with most of the scientists that this research is premature and unethical because there's too many risks and negative consequences? Or do you agree with Jin Kui that this is an important medical breakthrough that has a lot of potential? Give this video a like, subscribe to my channel if you're not, and use the comment section below to tell me your thoughts. And as always, thanks for listening.